Hello everyone, my name is Tanner Cartwright and this poster is about PCR. And I chose PCR because I think it's just such a beautiful tool and invention. Its simplicity and its applications are absolutely incredible. I've always been fascinated with it since the day I learned about it, and it was really cool to be able to do it in the lab. Introduction. PCR, or polymerase chain reaction, is one of the most important tools in microbiology. It was first developed in the 1980s and made it possible to take a tiny piece of DNA and make millions of copies of it. Before PCR, doing anything with DNA was slow and required a lot of material. This one invention changed everything, from diagnosing diseases to solving crimes. The goal of this poster is to look at how PCR works, where it came from, and how it's been improved over time, and what it's used for today. I'm pulling information from multiple studies and reviews to show how PCR fits into the bigger picture of microbiology and how it's still evolving. So here in Figure 1, we have a couple of different types of PCR. The top one is just, I believe that would be traditional PCR. The middle one is qPCR, or real-time PCR, and the bottom one is the modern sort of advanced method of PCR. There's much less steps in certain laboratories. That they've made it very simple over the years, which I'll talk about. Literature review and methods. Bartlett and Stirring, 2003, walked through the early days of PCR, using Taq polymerase, heat cycling, and gel electrophoresis to visualize results. Garcia, 2005, describes how PCR quickly found its place in clinical labs and became a standard tool in genetic testing. More recent work by Singh, 2016, talks about real-time PCR, qPCR, which allows scientists to measure DNA in real time using fluorescent probes. Donald et al. 2024 focuses on how PCR is used in forensic science to analyze STRs, short tandem repeats, and identify people, even from tiny or degraded samples. These studies all used similar basic equipment, thermocyclers, primers, DNA templates, but the techniques and goals varied a lot depending on whether they were focused on medicine, forensics, or research cloning. All of the references I used were sort of reviews and sort of historical analysis, and also sort of a rundown on applications in different fields, such as just biological scientific research and forensics. Alright, so the key findings from the literature. All sources agree that PCR completely changed how scientists work with DNA. The original technique was powerful, but pretty manual and prone to error. Still, it laid the foundation for everything that came after. The review by Singh in 2016 shows how much more sensitive and precise qPCR is today, especially for things like measuring viral load or gene expression. Meanwhile, forensic scientists have adapted PCR to work even when DNA is scarce or damaged. Kick et al. 2023 show how PCR is still crucial in modern cloning, helping researchers build plasmids and test if they inserted genes correctly. Across the board, PCR has proven to be incredibly adaptable. It just keeps finding new uses. So yeah, across the board, researchers agree PCR is a game-changing tool in biology. Before it, we didn't really have a reliable way of amplifying DNA so that we can study it, sequence it, do all the stuff we do in modern times. So really, without it, our field would be very different. Critical analysis. Even though PCR is amazing, it's not perfect. Some of the older methods were really sensitive to contamination, and early versions didn't have great controls or quantification. Even now, primer design can be tricky. If you pick the wrong sequence, you can get false results. Donald et al. Point out that forensic labs have to take extra steps to avoid contamination and misinterpretation. The newer methods like qPCR have improved a lot in terms of speed and reliability, but even those can run into problems if the reagents aren't handled properly or if the fluorescence is misread. So while PCR is a cornerstone technique, it still depends on good design and careful lab work. Yeah, so there's a few examples of when researchers had had sort of contaminated their research. Like there was one example. I can't exactly remember where I heard this from, but I'm pretty sure it's true. there were people who were trying to figure out if, like in Jurassic Park, like if you can take DNA from dinosaurs and sequence it. Turns out we know now very well that DNA does not survive that long, nowhere near that long. But I want to say it was back in the 70s or 80s. There was a research lab that came out with a paper that said that they had sequenced dinosaur DNA, but it turned out later on that it was a hair sample from either a researcher or one of their pets had fallen into the machines. So there's a few funny cases like that of sort of not user error, but just very sensitive methods like PCR. And then here we have a little diagram at the bottom of normal traditional PCR versus qPCR, real-time PCR. And really the difference between real-time PCR is you can just see what's happening using the fluorescence added into it. And then over on the middle right, we have figure 3, and that's just a picture of the thermocycler we used in the lab. I took that photograph myself. Implications and applications. PCR is used everywhere. In medicine, it helps diagnose infections like COVID or HIV. In agriculture, it's used to detect GMOs. In forensics, it helps solve crimes with just a few cells of evidence. Garcia 2005 shows how PCR gave scientists a fast and accurate way to detect diseases and mutations. Now we even have field kits that can do PCR outside the lab, making it useful for things like environmental monitoring and outbreak tracking. As technology keeps improving, PCR might get combined with CRISPR or AI-based primer design to make it even more powerful and specific. So yeah, there's a lot of avenues to improve PCR, making real-time PCR more simplistic. There's many companies that will, put all their reagents together, the primers and all that sort of stuff, to simplify as much as possible. Even though it's already, really a simple method of just increasing and decreasing the temperature with the right ingredients in the tube. So, PCR is used everywhere. I thought it was really cool in the lab. I did the in-person version, but they showed us the little PCR machine that they sent home, which I thought was really cool. I didn't know that it even existed, but I suppose now, knowing a little bit more about it, it's really not that complicated of a thing. It's more just knowing what you're doing. Conclusion. PCR has completely transformed the way we study DNA. What started as a simple method to amplify genetic material has become one of the most versatile tools in modern biology. It's used everywhere, from diagnosing diseases and solving crimes to cloning genes and monitoring the environment. One of the most impressive things about PCR is how well it has adapted over time. Even as technology has moved forward, PCR has kept pace, evolving into real-time methods, becoming more precise, and showing up in portable devices and field kits. It's still based on the same basic principles, but the ways we use it now are far more advanced. Looking ahead, PCR will likely keep evolving alongside new breakthroughs in AI and synthetic biology. It's hard to imagine any future in microbiology that doesn't involve some version of this technique. PCR isn't just a tool, it's the backbone of molecular biology, and it's probably going to stay that way for a long time. Alright guys, thank you so much for listening and looking at my poster, and hope you enjoyed my presentation.